Hello and happy Wednesday, everyone. So just a little friendly reminder that we are a week and a half away from midterm number 1. I'm going to talk about it a little bit more on Friday, but just a reminder that it's coming up. And I wanted to show you one thing that I've been working on that will hopefully help you with your midterm. So if you go over to Moodle, and if you scroll down on Moodle, hopefully you've been taking a look and seeing what's here. So there's all the lecture notes. If you go down here, midterm review, I have a sample midterm right here. And just now I posted some practice questions. So I'll just let you know what the practice questions are. Through the textbook, I get a test bank. And usually for midterms, I write all my own questions. But in this case here, I went through every chapter that we've covered and grabbed from each chapter a few questions, right? So I think I have 70 multiple choice questions there. So you can go in and it's just some extra practice questions for you to do while you study. You can do the practice test as many times as you want. It's not for marks, but just for studying purposes. I know some people struggle a little bit with the multiple choice. So I tried to choose questions that were things that we talked about. There may be a few things in there that have things like words and whatnot we didn't use. But you should be able to do all those questions with a little bit of studying. Obviously, some of those things we haven't talked about yet, but that's a resource for you to look at over the next week and a half as you prepare for the midterm. So like I said, I'm going to talk about the midterm on Friday in a little bit more detail. It'll be a regular lecture on Friday, but I will spend about 5 minutes kind of giving you an idea what to expect for the midterm. So you can look forward to that on Friday. So a week and a half away. One other reminder is that our first lab assignment is due tomorrow, and that is due by the end of the day. So if you have not handed that in, please hand it in by the end of the day tomorrow, and then we're going to work on Lab 3. One more quick note. Lab 3 is a formal report, and so it's a big major assignment. It's worth about 14% of your final grade. I'm going to give you 3 weeks to do it. Normally it's 2 weeks, but I'm not going to have you hand it in during the reading week, so you get an extra week to do it. And so what I'll be doing starting Friday as well is spending a couple of minutes at the beginning of every lecture giving you some lab report tips. So it'd be good to tune into that and take a look at what I'm talking about for writing your lab report. So more on that later. Today, we are going to be talking about prokaryotes. And so last day, we had made a table that compared prokaryotes and eukaryotes. And so today, we're going to get into some more details on prokaryotes, which include both the bacteria and the archaea. So here's some numbers for you. I don't think people necessarily know how abundant these things are. So you're probably looking at these numbers and wondering what they are. So here is the first number, 5,400. So that's about the approximate number of species of mammals that scientists have classified. And crazy thing about that is about 1,000 of those species are actually different types of bats. So interesting. 10,000. One spoonful of fertile soil is going to have billions and billions of microbes in there, most of them prokaryotes, and representing about 10,000 species. So how many species have we actually characterized? Somewhere about 4,000 to 5,000. There are many more than that, and we can see them and we can detect them through genetics techniques and things like that. But most of them out there, in fact, some people believe there's about 4 million species. Most of them we haven't been able to characterize, which means we haven't grown in the lab, or maybe we can't grow them in the lab. So these things are really abundant, and they're all over the place, these prokaryotes. So a couple more numbers for you. 30 to 40 trillion. That's about how many cells you have in your body. And 30 to 60 trillion. That's about how many cells that are bacterial in your body. So most of those, by the way, are in your intestine, but we do have bacteria on our skin, in our nose, in our mouth, and all sorts of other places on the body. So, if you think about your human body, we've got about just as many bacterial cells as human cells. And so you know, that makes us kind of some interesting things going on. You can think of your entire body as an ecosystem that's harboring all sorts of microbes. So I think what I'm trying to say is this: they're everywhere. Almost everywhere we look, we find prokaryotes, and that includes the bacteria and the archaea. They're very, very abundant, very, very diverse, and there's lots that we don't know about these things, and we're learning about them all the time. We're going to talk a little bit about how some of them cause disease, so that's probably not going to be quite today's lecture. A little bit more on next day's lecture, but they're very important for that reason, because many of the diseases that we get are actually bacterial in nature. So last day, I had made that table comparing the prokaryotes and the eukaryotes. I threw down a few words in terms of, you know, what's going on with the organization of the DNA and the ribosomes and things like that. And so today we're going to start talking about a few of those things. Next time we'll eventually start talking about eukaryotes. So that will be probably on Friday. But today anyway, we're going to focus on prokaryotes. And so just a reminder that we talked about the 3 domains of life. And so the one domain was the eukaryotes. And the prokaryotes, there were 2 domains. We had the archaea and we had the bacteria. So we're going to focus mostly here on the bacteria. And there's a few reasons for that, and I'll get to that in a minute. But what I want to do is just talk about the archaea for a minute. And I'm going to make some notes about the archaea. So I'll get to that in a moment. So you can see this, this is a photograph of a hot spring somewhere. I can't remember where this photograph was taken. I didn't take it. And if you go there, you can measure the temperature, and the water it's almost 100 degrees. It's something like 95 degrees, which is too hot for pretty much anything to live. But if you put a glass test microscope slide in there, and you leave it for a day and come back and stick it under your microscope, you might find some organisms. And those organisms are going to be archaea. So some of these are extremophiles, meaning they live in these extreme environments. But this is not the case. This is sometimes, if you find older textbooks, they will say that all archaea are extremophiles, and that is not true. We find them pretty much everywhere we look. They're found in the human body, they're found in the soil. They just grow really slowly, most of them, so they're not as easy to detect as bacteria. One notable exception of something that is easy to grow and easy to detect are these ones here. So you can see there's a little cartoon, and she's telling her friend about her movie. She's halfway through a sentence, and then doot, and it says, "Don't be embarrassed. It's not your gas, it's your archaea." So archaea are organisms that make methane. So methane is part of your flatulence, and that's what's going on there. So I wanted to make some notes about the archaea, and then probably I won't really talk about them too much again, because like I said, most of what we know about prokaryotes is due to bacteria, and I'll give you one good reason for that in a moment. So I'm just going here, and I'm just going into Word, and just going to make some notes on the archaea. So you can see the very first note I have is that they are prokaryotes. That means they have no nucleus. So if you remember last day, I had told you a couple other things about the archaea, is that they have a cell wall, and it is not peptidoglycan. Maybe it's
we can't see what you're typing right now.
Oh, you can't see what I'm typing. Okay, well, thank you for that. I guess I forgot to share my screen. That's better. They do not have peptidoglycan, but maybe it's better to tell you what they do have. They may have pseudo-peptidoglycan. So remember, that's a different carbohydrate, and it kind of looks like peptidoglycan if you're not really taking a good look. It has different glycosidic linkages, is a big part of what it has. And some have something called S layers. So that's our first note, and that's one difference of these things. Another difference is they have unique phospholipids. So remember that some have monolayers. I showed you a diagram about that before, layers, and there's some other unique chemistries around their phospholipids. What else can we say about archaea? This is kind of an important one. There are no known pathogens. So I put that in bold because this is a major reason why we have a lot less research dollars to study them. None of them seem to make us sick. So there goes all the medical research that is not going to happen with archaea. As I mentioned, they tend to grow really slowly as well. And so, you know, if you want to grow a culture of E. coli, you can grow it up in a day very easily. Some of these archaeal organisms will take 6 months to grow. So that doesn't really make the research very easy. So that's another reason why we don't study them so much. There were a couple other things I was going to say about them. I guess I should have put this one first. They are found, we'll say, everywhere, meaning they're found pretty much in every place on Earth that living things would live. They're not going to be found on the moon or something like that we know of. Who knows? Apparently we're going back to the moon. They're found everywhere. Some of them are extremophiles. So that's meaning they're living in extreme environments. And so an example of that is these hyperthermophiles. Hyperthermophiles, so meaning they live in very hot places. Some of them live in really salty places like the Dead Sea, which has a really high salt concentration. And some of them are methanogens. So these, methanogens. So they make methane, which in some people, they make more than others. And if you're a cow, they make way more in cows than they do in humans. I feel like there was one more thing I was going to say about them, but oh, maybe I will just say the last thing is they have many unique biochemical properties. We'll just leave it at that. So if you take a look at, for example, little things like how their DNA is organized, or you know, what kind of enzyme they have for an RNA polymerase. It's different. It's unique for the archaea than it is compared to the other triggers. So that makes them unique in that their chemistry is different in many places, and so they, you know, we've given them their own actual group. Okay, so hopefully you got all that. Like I said, I'm going to talk about prokaryotes, and most of what I'm saying, unless I'm specifically saying bacteria, do apply to both archaea and prokaryotes. But a lot of that, like I said, is really more referring to bacteria because we know so much more about them. All right, so back to the PowerPoint. All right, bye-bye archaea. It's too bad we don't know more about you, but maybe eventually. So there's my note. It says they're found all over the biosphere. There are no known pathogens. So we're not really going to talk about them too much in this class. It shouldn't say they will not be covered, but they'll be minimally covered, I guess I meant to put. So what about bacteria? Okay, they are their own group, and what do they do? So they live with us. There's E. coli, it lives in your intestine. We'll be talking about E. coli a little bit more next day. Some of them make us sick. This is Yersinia pestis. This is the cause of the Black Death. And that's a little diagram on the right is a plague doctor. So this one makes us very, very sick. And there are many that can make us sick. Some of them are part of the biosphere in that they make oxygen for us. So these cyanobacteria make a huge amount of oxygen in the atmosphere. And so we like oxygen, so that's good. They're involved in all sorts of decomposition and nutrient cycling. What I'm showing you here are these nitrogen-fixing bacteria that are found in the roots of legumes, and so they're bringing nitrogen, they're part of the nitrogen cycle. And many, many, many other things bacteria are doing. They're digesting cellulose in cows. They're involved in yogurt making. We use them for biotechnology. They actually help us to develop our immune system when we're babies. So they do lots of interesting things. And it's unfortunate we can't spend too much time on them, just really 2 lectures. So what we're going to talk about today is a lot of these parts that you might see in a bacterial cell. And I'm going to make a little table as we go along. I'll kind of update a little bit. I'm not sure if we're going to get through all of them today, but you probably are familiar with the components of a eukaryotic cell, at least some of them. You know, we've got the nucleus, we've got the Golgi body. So we don't have any of those organelles in bacteria, but they do have a lot of features, and it's important to talk about them because a lot of these features actually help them to live in their environment, and then if they're a pathogen, they might help them to you know, make somebody ill. So it's important to kind of learn a little bit about these things. So let's start off and talk about the cytoplasm. So what is the cytoplasm? The cytoplasm is the part inside the cell that contains everything, right? So inside the membrane, and you've got often a lot of people think of the cytoplasm as just water, but I'm showing you this diagram here. This is an artist. He's also a biochemist, and he tries to draw things in such a way to show you some interesting educational kind of details. And one thing I really like about this diagram is he's showing you that the cytoplasm is really loaded with all sorts of things. In fact, each of those things he's drawn there, they're not just purple blobs. He's actually using accurate models on these things. So let's take a look at what we have here. So the first thing to note, one thing that's in the cytoplasm are the ribosomes. And you can see that ribosome there has a little messenger RNA on it. And ribosomes, of course, they make proteins. So let's talk about ribosomes. So first thing to know, I told you last day that prokaryotes have smaller ribosomes than eukaryotes. And you can see there's this number here. We've got 70S and 80S. So S, you can think of S as size or sedimentation. It actually stands for Svedberg. So Svedberg was a guy, sounds Swedish maybe, and he was studying ribosomes. And so they named the unit after him, right? So he was using centrifuges to see how fast things would pellet in the bottom of a centrifuge tube. And so it turns out that, that bigger things, they pellet faster, and they get a larger number in terms of what he's talking about. But what really matters is you have different ribosomes. And so it turns out prokaryotic ribosomes are a little bit smaller, and these 70S ribosomes, you can break them into two parts. We have this thing here is called the large subunit, and this one here is called the small subunit. And so when you look at these, the small subunit is 30S in prokaryotes, and the large subunit is 50S. So notice it doesn't add up, right? Because when you separate things, they sediment differently, right? So it's not necessarily a mass, it's how fast they sediment in a test tube. The eukaryotes, the large subunit and the small subunit are bigger, and you can break those things down, and they are made out of proteins, and they're made out of RNA. So you can see rRNA means ribosomal RNA, and the components are different. They do the same thing. So ribosomes, of course, are involved in translation. So translation. And translation is protein synthesis. So they do that by reading the messenger RNA. And we're going to talk about that way later. I think it's topic 15 or something like that. So we'll get there eventually. So prokaryotic ribosomes are different than eukaryotic ribosomes. So there's our little ribosomes. There's actually a transfer RNA. That's something else that's going to be involved in the translation process. And he's got a bunch of enzymes in there. So if you read his description, he tells you what each of these enzymes are that are found in the bacterial cell. This guy also has some DNA, and if you look carefully, he's got DNA polymerase there. And DNA polymerase takes one strand of DNA and makes two daughter strands. And you can see they're being synthesized there by the DNA polymerase. So pretty cool. So let's talk about the DNA now. And so there's no nucleus. So a nucleus is a membrane-bound organelle. But the DNA will often clump, particularly when seen under an electron microscope. Electron microscope, I think I mentioned last time, in order to, prepare the specimen, you're going to dry it out. And during that drying process, the DNA will clump, and it can be very visible under an electron microscope. So sometimes they give it a name and they call it the nucleoid or the nucleoid region. And so what they're just saying is that there's no nucleus, but the nuclear material, which is the DNA, is found there. And you can see on this one here, it's actually seen very, very clearly. You're actually looking at, this is a dividing cell, by the way. So it's replicated its DNA, and so it has two nucleoids because it's replicated its genome. So it has two copies. So one thing to know about the bacterial genome is that most bacterial genomes are a single loop of DNA. So you can think of it as a giant, not necessarily a hula hoop. You know, hula hoop would be a big circle like this. You know, it's a bit more like, you know, twisted up and folded up. And I'm not exactly sure what pattern it folds up, but it's a little bit more like that because it's got to be able to fit inside the bacterial cell, which is a very, very tiny thing. So one more thing to say about the DNA, and then I'm going to make a couple of notes for you, is that some bacterial cells have these little tiny pieces of DNA, and they are called plasmids. So you can think of a plasmid as almost a mini chromosome, and very, very mini. So a typical chromosome in E. coli will have maybe about 4,500 genes. A typical E. coli plasmid might have 3 genes. So very, very different in terms of the size. So they're kind of like little extra genetic elements, right? So they've got just a few spare genes. I know I have a table here that talks about some of the things that are found in plasmids. So this is the big one that scientists are always worried about, antibiotic resistance. So it turns out some of these plasmids out there actually carry the genes to give antibiotic resistance, so drug resistance to pathogens. So we're hugely concerned about this. And these plasmids can be shared between different species. And so sometimes this is a really, really big concern. There are other things on this list. You can see Apilus, you don't know what that is. Toxin production. And so sometimes these plasmids contain an extra gene or two that kind of help them out in their environment. And that's kind of what plasmids are all about. So I'm going to switch now and make some notes. Like I said, I usually do this on the whiteboard while I'm talking, but not so easy to do with this online thing. So you can see I'm making a table, and you can see I have structure. So we can maybe put structure as the nucleoid, and nucleoid, right there we go. And the composition. So what is the nucleoid made out of? It is made of DNA, and we'll call it plus associated proteins. There are lots of proteins involved with the DNA because, of course, there's proteins that are synthesizing new DNA, and there are proteins that are organizing it, and so on. So I'll just say it's mostly DNA, a little bit in RNA that's being produced, and associated proteins. So what's the function of the nucleoid? So by the way, DNA plus associated proteins can also be called a chromosome, right? So you probably know that word. So what's the function? Contains the genetic material of the cell, okay? We also talked about plasmids. Okay, what are plasmids? And plasmids are small circular DNA molecule. What do? They carry an extra gene, carry a few, let's just say a small number. So typical, like I said, is anywhere from maybe about 3 to 15 genes. There are some with less, some with more. 3 to 15 genes, and I'll just say such as antibiotic resistance that can help the organism survive in an environment. Okay, I think I forgot to put ribosomes. I was going to put those first, but I guess the order doesn't matter. So by the way, composition is to kind of help us remember what these things are. That's what I think about. I think about the macromolecules and what these things are made of. So ribosomes, remember ribosomes are made out of RNA, and it's called ribosome RNA, ribosomal RNA, and they're also made out of proteins. So what do? They carry out translation, which is another word for protein synthesis. There we go. So maybe I'll put in, okay, obviously I cannot spell today. Hopefully that will improve. So what does this mean? It means they take the information from messenger RNA and make an amino acid chain, which we also call a peptide, or a polypeptide if it's a big one. Okay, so I will come back to this. If you didn't get all that, don't worry, I'll come back to it. And like I've said before, if you really want, you can always just ask me to email this to you as well. So back to bacteria and all their interesting structures. Okay, so there is, back to David's diagram here. You can see the cytoplasm is loaded with all these goodies, ribosomes and DNA and enzymes and those kind of things. And notice he has a, this is actually E. coli, and E. coli is a gram-negative organism. So remember our cell wall in gram-negatives, we have two membranes. So we've got an inner membrane, which is shown here. And we've got an outer membrane, which is shown here. And in the middle, we have our peptidoglycan. So you can see that he's trying to show those structures. And if you look carefully at those membranes, we have phospholipids and all sorts of peripheral and integral membrane proteins. So nice diagram there. So I guess I thought I was going to talk about peptidoglycan, but maybe that's coming up here in a minute here. So one thing to think about bacteria, of course, is that they're small. They have one chromosome. So when they divide, they replicate. There's no mitosis. You don't need mitosis or meiosis when you've got one chromosome. If you've got lots of chromosomes, it's really important to organize them, and you've got all those spindle fibers to make sure every cell gets a fair allotment of the chromosomes. It's a lot simpler when you are a prokaryote and you have one chromosome. So this has a name, binary fission, right? So binary means two, and fission means divide. So binary fission just means divide in two. I know it's pretty lame, and in fact, in a lot of the scientific literature, we don't even use the term, we say bacterial division. But anyway, it's the term that's used, and so it's a good one to learn because it's technically correct, even though if it's not too fancy. So how does this work? You can see here we have, up here we have a cell, and you can see in the second image, it's starting to replicate its chromosome. So we've got one circular chromosome here, and it's just starting to replicate there. You can see the two little green parts where it's just starting to separate. And notice this note about it attaching to the membrane. So take a look at this. It's attached to the membrane, and this is actually how it pulls its two chromosomes apart. So the cell is going to stretch out, and as the cell stretches out, one chromosome is attached to the cell on the left, and one chromosome is attached to the cell on the right, and it elongates and basically yanks them apart. So pretty cool. We don't need any spindle fibers. We just need to attach our chromosomes to the cell membrane, cell wall, and it's all good. The last step is that they have to kind of do some pinching here. And so the membrane kind of gets pinched together, it separates, and of course a cell wall is kind of made in between in the middle. So I have, oh, I didn't know this had volume. There, we try that. Anyway, you can see there's a couple of videos here, and you can see we've got E. coli, which is a rod-shaped organism, and we've got Streptococcus, which is a coccus, which is a round-shaped organism, and they're dividing. These are time-lapse. They don't divide that fast, and you can kind of see them stretch out, divide, stretch out, divide, stretch out, divide, and that's how they do the job. So I'll come back to that in a second, hold on. So we are going to talk about mitosis and meiosis, by the way, and that is topic 12 or 13. So we'll get there eventually. You probably remember there's some phases, there's telophase and metaphase and so on. So we'll get there, but for this time, you should know binary fission when we want it, because we are talking about bacteria. So I do have kind of this little test yourself thing here, true or false. I don't know what happened to that yellow box. It kind of shrunk funny there. So let's go through each of these and think about this. So first one, it says bacteria divide by binary fission and archaea divide by mitosis. So this is true, this is false, right? Archaea are prokaryotes. So archaea divide by binary fission. All right, number two, the nucleoid region is surrounded by a membrane. No, of course not. Why would we say that? The nucleoid is in a prokaryote. There is no membrane-bound nucleus. Number three, prokaryote ribosomes contain 40S and 60S subunits, and this is also no, that's too big. It was 50 and 30. 50S and 30S. Okay, so I threw this out because I don't have a Kahoot for you today. I'll have one for you for next day, and we'll get a chance to review some of these bacterial structures. Okay, so let's talk about that cell wall. So this is just a review of the cell wall. And remember, the cell wall is made out of peptide glycan. And remember that there's two types. So maybe I will go back and we'll make a note about that in our table. So structure is the cell wall. And the cell wall is made out of peptidoglycan. And what is the function of the cell wall? So the cell wall gives the cell shape, and I guess we'll say strength. I don't know if that's quite the right word, but and helps it resist osmotic pressure. So that means that a bacterial cell, really can't spell today. You know, it's hard to talk and write at the same time. That means a bacterial cell is like a plant cell in that you can give it water and it's not going to burst open. And that's what I mean when I'm talking about osmotic pressure. So let's take a look at a little bit more information on gram stains for you. And we are going to talk about them in next week's lab, so a week from now. So it's important to know a little bit about them. If you remember last day, I was talking about the gram stain and the gram-positive cells. They trap the crystal violet, that's purple stain, and they turn purple. And the gram-negative cells don't. And so in the end, they stain pink. So how does this stain process work? So if you take a look, here's some cells, and they are you know, not very colored. You can barely see them on a slide if they're not stained. And so the very first step, we add crystal violet. So we'll call crystal violet CV. And it turns out that the crystal violet is positively charged. So this is going to help us out a lot because the second step, we're adding iodine. So iodine, it turns out, is negatively charged. So the iodine, and there's my iodine and my crystal violet. So the iodine and the crystal violet, they kind of just form these complexes. And remember that the gram-positives have that big, thick peptidoglycan cell wall, and it kind of gets trapped in there. The gram-negatives have that extra layer, that extra outer membrane. And so when you do this washing step, which is step number 3, the crystal violet just dissolves away, but it remains trapped in the gram-positive cell. And so lastly, what you do at this point, you can see the gram-positives, we can't see the gram-negatives, so we throw in a counter stain, and the counter stain is called safranin. Sometimes we call it safranin red, which tells you what color it is. And so, you know, what happens when you add something that's pink to something purple? It just looks purple. So the gram-positive stays purplish looking, but the gram-negative cells pick up the pinkish reddish stain. So that's how gram stain works. And so I'll go over it again next week in lab 4, but I thought it'd be worth it to kind of mention it now. There's some images. I think I showed you some of these ones before. There's anthrax on the left, and it's gram-positive and purple, and kind of looks like spaghetti. And there's Staphylococcus aureus on the left as well. And you can see Staphylococcus, remember, coccus is round, and staph means cluster. So you know, these clusters of cells that kind of look something like that. On the right, we have E. coli. That's not a very good E. coli image. I'll have to get a better one for you. I just found that on Google image search. It's a little blurry, but you can see that E. coli is a nice rod shape. So a rod shape, a word for that is bacillus. And on the bottom, we have Neisseria gonorrhea. So gonorrhea, they are little round cells, and often they are found in kind of diplococcus, which means there's two together. So this is used, the gram stain is actually really important for diagnostic purposes. And it's more than 100 years old, and we still use this today when people get samples. You know, they have an infection, and in this case here, a person has gonorrhea, a quick gram stain, and they can tell you right away that it's gonorrhea kind of thing. Doesn't work for everything. There's a lot of things that look like E. coli under a microscope, but if you see anthrax, you certainly don't want that one. So just another reminder about the biology of the gram negatives, in that they have that outer membrane, and that outer membrane has this thing here, which is called a lipopolysaccharide, which usually we just call LPS. And the lipopolysaccharide is something called an endotoxin. And these things, these endotoxins, they will give you an immune reaction. Gives an immune reaction. And this is why we do not want blood infections with bacteria, because that immune reaction in some cases can be really serious, and in extreme cases lead to death. So the other feature of the gram negatives that I talked about last day was this periplasm. So this is kind of a little zone, a little compartment between the two membranes. It's very, very tiny, but sometimes it has enzymes in there, and enzymes that will resist drugs, so they have for antibiotic resistance. So it makes gram negatives a little bit harder to treat sometimes. So one thing to note is when you talk about bacteria, most of the species out there that we talk about are gram negatives and gram positives, but there are other groups out there. And so I just wanted to show you that there is another important medically relevant bacterium that is not a gram negative or a gram positive. It's actually called a mycobacterium. And you can see the organism that we're looking at here is called Mycobacterium tuberculosis. So this causes tuberculosis, which is a pretty significant human disease. It's a lung disease, and it's pretty significant in some parts of the world. So they have a unique cell wall that's called an acid-fast cell wall. And it's kind of given that funny name because of the staining procedure. It involves acid and whatnot. And I'm not going to get into that, but you can see it has a different structure, right? It's got some peptidoglycan, and it has another carbohydrate called rabinoglycan, and then it has this other thing called a mycolic acid. So mycolic acid is actually a really waxy substance, and so if you try to do a gram stain, the wax just repels the stain. So there's a different procedure called the acid-fast stain, and it uses different different dyes, and it stains most things blue. So you can see in that diagram there, most of the background is blue, and you can actually make a lot of human cells here, right? So this is all human tissue that we're seeing here, and the mycobacterium stains kind of this pink fuchsia color, which you can see in that by that little arrow there. So just for your information, there are other types of bacteria besides gram negatives and gram positives. Just most of them do fall into those two categories. Okay, so here's another little test ourselves thing that we can try to do, and this is involving types of cell walls. So this one here, I went out of my way to word it a little tricky. I usually try not to have things so tricky on a test necessarily, but I just want you to think about what is going on here, right? So let's take a look at these, right? And we'll start off with the animal cell. We know an animal cell doesn't have a cell wall. So what does an animal cell maybe have? It maybe has an extracellular matrix. So that is actually one of the answers. So we can connect those two. Okay, so let's take a look at these other answers here. So two membranes, chitin, animals cannot digest this, stains purple. Okay, so there's some good clues here that we can work with this. So hopefully you remember which one stains purple. That would be the gram positive. What about chitin? Hopefully you remember that chitin is found in fungi, the fungal cell wall. So there's another one. So narrowing it down, two membranes. So there was only one system we talked about that had two membranes, and that was the gram-negatives. So there we go. So just two left. Animals cannot digest this and never peptidoglycan. So those are, I know, funny clues, but I'm just trying to get you to think about these things we've talked about. So plants are made, their cell walls are made of cellulose. And remember that animals cannot digest cellulose. And animals that eat cellulose, it's actually the bacteria in their guts that are doing it for them. So lastly, that leaves the archaea, which never have peptidoglycan. They are prokaryotes. They might have pseudo-peptidoglycan, but not normal peptidoglycan. So that's kind of the rationale behind that. So just a quick kind of let's test ourselves and think about what we've talked about cell walls. Okay, so moving on. Let's talk about a few other bacterial features, and we'll see how far we cover today. And like I said, I was assuming I wasn't going to get through it all today anyway. But there's a lot of other bacterial features that are important aspects of the cells, and one of them are these protein things. And these proteins have different names. We use the word fimbriae or pili. Those terms are used interchangeably. I don't know why we have two words for it. But I guess, you know, it's just kind of the way it works sometimes. And so you can see that it says these are attachment appendages made of proteins. So you can think of them as little sticky fingers. And so bacteria, you know, they're living out in the environment, and sometimes it's handy to grab onto things. So it could be a rock in the environment, or a piece of sand, or a plant, or in the case of pathogens, these could be attaching to human tissues. So if you've ever had a bladder infection, that's because these things are attached to human cells up in that plumbing in your body. And if they don't have the pili, they can't attach there, and that species can't give you an infection. So these are really important for pathogenesis. Here's another picture of one. Like I said, you can think of the little fingers or little hairs, and they're very sticky. So these words are used interchangeably, fimbriae and pili. So you should know both of them. You may have noticed from the last slide, I have this note about there is one special unique kind of pili called a sex pili. And that's worth it to talk about too, because they do something kind of interesting and unique. So I'll show you a picture of that one. So this is a sex pilus. So pilus is one, pili is many. Okay, it's just a singular plural kind of thing. So what are we seeing here? This is an electron micrograph, and there's something attaching these two cells. And so what you're seeing is these two cells in an intimate moment, and they're having, it's called a sex pilus. So they're having sex or conjugation. So what does that mean? It means they are exchanging DNA. That's really what sex is. It's exchange of DNA, and in a biological sense anyway. So no reproduction going on here, and it's just exchanging of DNA. So the term for that, the technical term is conjugation. So if you've ever watched like a movie and they have there's a prison, and they've got like somebody is, their wife or husband is coming to visit, and sometimes they have conjugal visits, and that's so they can be intimate once in a while. And that's so same word, conjugation, conjugal, right? And it means sex. So I just want to show you how this works. So if you take a look at these cells I have at the top, one of these cells is F positive. So this is geneticists. They love to name things in weird ways. And so F means fertile. It means it has the gene for fertility, which is anyway, it works, I guess. And so this other cell is F negative, meaning it's not fertile, doesn't have the gene. So what is the gene? It is found on a plasmid called the F plasmid. So at least they are, being consistent here. And so you can see that the donor cell, or the F positive cell, somebody needs to have the DNA in order to make the gene to make the pilus. But the other cell doesn't need it. The recipient cell does not need to have that plasmid. And here is the crazy thing, is they don't even have to be the same species. So unlike species can do this kind of thing and exchange DNA. And this is why we're, always worried about antibiotic resistance, because they can share it between different species, which is kind of scary. So how does this work? The donor cell makes a pilus. Like I said, these things are made out of proteins. And there's one textbook I read that kind of related to a grappling hook. So that's actually a pretty good description, because what happens, I'll show you the next step here, is the donor cell actually brings the recipient cell closer. So you can only imagine if human intimate moments were so dramatic. If the men around here went around with grappling hooks, it would be kind of interesting or scary or a little bit of both. But that's how they do it. They pull them in, right? And then the magic happens, which is the DNA exchange, right? So you may know that DNA is double-stranded, and it's got a plasmid. And so what it does is it spools off one strand. I don't really know how this works, but somehow it spools off one strand, and it's believed that strand goes, it's believed that the pilus is hollow. We don't actually know, we've never actually proven that, but that's the belief anyway. And so one strand will go through the pilus, and now they both have a strand, and they can, make it into double strands. And now both cells, you can see, are fertile or F positive. And so whatever else was on that pilus, now they have, the new cell has the ability to make a pilus. And if there's any other genes on that plasmid, such as antibiotic resistance, now it has it. So conjugation is a big deal in hospitals, right? If you have one organism that has antibiotic resistance, it can spread it to more dangerous germs. And so this is something that we think about all the time when we're tracking different strains of bacteria that are causing people to be sick. Okay, so maybe it's time we make a note about these. Okay, here we go. So we have Fimbriae, or pili. Remember, pilus is one, so I'm using the plural term. And these things are made out of proteins. And these are for attachment to surfaces. And we also have the sex pilus, and that is used for conjugation. So it's attaching to another cell, but has the specialized purpose of transferring the DNA. Okay, a couple minutes left, and I'm going to talk about one more structure here. And so you're learning some F words today, some good F words. So Fimbriae, fertile, and I forgot to mention this one. This is a video that was captured of two cells conjugating, so using fluorescence. This is actually very, very recent, so kind of interesting. So one more F word I want to teach you today, and is flagella. So what does a flagella do? A flagella helps things to move around. So when we're talking about movement in microorganisms, the word we use is motile. So if you take a look, some of those, maybe some of those are dead, but others are kind of moving around. And how are they moving around? They're moving around using a flagella. So we're going to talk about flagella a little bit more in after the midterm. I'm still on the Word document. Oh, you missed the video. Okay, I'll have to go back and play that for you. Hold on a second. 2 videos you missed. Thanks for letting me know. Okay. All right, here's the video of, play that again. There's the video. How come it's not playing? Oh, there we go. There's the video of the conjugation. So you can see the pilus going out like a grappling hook. So kind of interesting. Like I said, very recent image that somebody has done. So motility, there's the other video of bacteria moving around, and I'm going to finish on this slide today. I'll come back to it next day, because what I want to do is just make a quick note on flagella. So flagella, these are swimming appendages, and they're a lot bigger than pili. So I'll show you. Let me just go ahead here. No, where is it? There. I'll show you this image here. You can see the Fimbriae, or the pili, are a little bit more like hairs, and the flagella are a lot more like tails. So let's make a note on that, and that's where we'll finish off today. Bacterial structures. And you can go home and tell everyone you learned some new F words. And so these are also made out of proteins. Okay, and so flagella are for movement or motility. And so they're, like I said, kind of like big tails. All right, so last thing I'll say about this today is that flagella are also really important for pathogenesis, right? So it helps them move around. So you can imagine if they can move around, they can infect more tissues, they can get to more environments, those kind of things. So they're really important for some organisms in their pathogenesis. Okay, so that is it for today, and we'll come back next day. I'll have a Kahoot for you after we learn about a few more bacterial components, and I will also talk about the midterm on Friday as well. So I hope you have a wonderful evening, and I will see you on Friday.